Imagine a world where cancer treatments zero in on rogue cells like a laser-guided missile, sparing the innocent bystanders. That's the thrilling promise of a groundbreaking mRNA therapy just unveiled by scientists. But here's where it gets controversial. Could this smart technology be the key to curing diseases, or might it open Pandora's box for unintended genetic meddling? Stick around, because we're about to dive into the details that could change medicine forever. New York, New York. December 15, 2025. At the prestigious Icahn School of Medicine at Mount Sinai, a team of brilliant researchers has pioneered an innovative mRNA system that activates healing genes only in the cells where they're truly needed. This breakthrough, tested successfully in mice, might pave the way for more secure and accurate therapies against cancer and a host of other illnesses. Dubbed the cell-selective modRNA translation system, or cSMRTS for short, this engineered mRNA is crafted to respond specifically to certain cell types. The results of their work appeared in the online edition of Molecular Therapy on November 15, accessible at https://www.cell.com/moleculartherapy/s1525-001/25-00954-2, published by Cell Press. Building on the success of mRNA COVID-19 vaccines, which essentially turn cells into mini factories churning out protective proteins, the researchers aimed for pinpoint precision. Vaccines can produce proteins anywhere in the body without much fuss, but cancer care demands targeting only the tumor cells while protecting healthy ones. Traditional lipid nanoparticle delivery methods, you can learn more about them here. https://www.sciencedirect.com/topics/medicine-and-dentistry/nanoparticle, haven't always nailed this, often leading to unwanted side effects, as the team explains. Our vision was to overhaul mRNA therapies from the ground up, shares Magdalena M. Zak, Ph.D., the lead author and an instructor in the Cardiovascular Research Institute and Department of Genetics and Genomic Sciences at Mount Sinai. Currently, we expend enormous energy perfecting delivery systems, yet we still encounter plenty of unintended consequences. We flipped the script by designing the mRNA itself to detect its surroundings, essentially asking, am I in a cancer cell or a healthy one? If it's the wrong spot, it switches off automatically. This self-decision capability sets our approach apart. And this is the part most people miss. The elegance lies in how the system mimics a biological switchboard, responding to microRNAs, those tiny RNA molecules that fine-tune gene expression in cells. In their mouse experiments, the scientists evaluated cSMRTS across two cancer scenarios. The technology functions as an internal toggle, activated by the unique microRNA profiles in cancerous cells. Put simply, it employs two mRNA components, one that encodes for the enzyme Cas6, which chops up RNA, tagged with a marker that cancer-linked microRNAs latch onto. The second carries the actual healing gene, capped with a small RNA hairpin that Cas6 can snip. This clever arrangement empowers cancer-specific microRNAs to control the process. In tumor cells, these microRNAs bind to the Cas6 mRNA and disable it, freeing the therapeutic gene to kick into action. But in normal cells lacking those microRNAs, Cas6 gets produced and slices the therapeutic mRNA, blocking any activation. It's like a security code that only unlocks in the right environment. Delivered via standard lipid nanoparticles throughout the body, the platform demonstrated impressive precision. Gene expression surged over 100 times more in breast and colon cancers. It plummeted more than 380 times in vital organs like the liver and spleen. Tumors shrank by 45% with a gene like 10, which suppresses cancer growth. Combined with mRNA-driven immunotherapy, it reduced tumors by up to 93%. What's truly exciting, and here's where it gets controversial, is the system's versatility. Since it's built for cell specificity, it's not limited to one ailment or one treatment type. In theory, it could be tweaked for precision drugs tackling everything from cancer to autoimmune disorders and metabolic issues, says senior author Lior Zangi, Ph.D., profile at https://profiles.icon.mssm.edu/lior-zonai, an associate professor in medicine, cardiology, and genetics and genomic sciences at Mount Sinai. With 15 years of experience in mRNA for heart health, relying on direct heart injections, I'm fascinated by how this could enable safe, targeted gene activation in specific tissues or organs, without invasive methods or stray gene activity. Existing nanoparticle strategies mostly confine mRNA to vaccinations. By making the mRNA itself selective, cSMRTS aims to minimize risks and broaden mRNA's medical applications. For patients, this translates to potentially more effective, gentler cancer options, with prospects for treating other conditions down the line. The group has secured patents and is pushing forward with commercialization and further preclinical trials. Their paper, A Tumor-Selective mRNA System Enables Precision Cancer Treatment, credits these authors, Magdalena M., Zach, Jameen U., Alberto Utrero Rico, Wenk Walter, Gayatri Mainkar, Matthew Ajmi, and Anu Kurian, Ashikar Rahman, Daniel Lozano Ojalvo, Jordi Akando, Torsten Haferlach, Ramon E. Parsons, Philip K. Swirsky, and Lior Zangi. Funding came from a NantRNA research partnership and NIH grants are 01HL142768-01 and are 01HL149137-01. About the Icahn School of Medicine at Mount Sinai. The Icahn School of Medicine at Mount Sinai is globally acclaimed for its top-tier research, education, and clinical programs. As the exclusive academic affiliate for the 7 hospitals in the Mount Sinai Health System, the largest such network in the U.S., it serves New York City's diverse population with unmatched care. The school offers highly sought-after MD, PhD, MD-PhD, and master's programs, enrolling over 1,200 students. It boasts the nation's biggest graduate medical education initiative, training more than 2,600 residents and fellows across the health system. Its Graduate School of Biomedical Sciences provides 13 programs, spearheads cutting-edge research, and mentors over 560 postdoctoral fellows. Ranked 11th in NIH funding nationally, Mount Sinai places in the top 1% for research investment per investigator, per the Association of American Medical Colleges. Over 4,500 experts collaborate across departments and institutes, focusing on translational research that bridges lab discoveries to real treatments. Through Mount Sinai Innovation Partners, MSIP, they accelerate the commercialization of medical advances. Mount Sinai Health System member hospitals, the Mount Sinai Hospital, Mount Sinai Brooklyn, Mount Sinai Morningside, Mount Sinai Queens, Mount Sinai South Nassau, Mount Sinai West, and New York Eye and Ear Infirmary of Mount Sinai. About the Mount Sinai Health System. The Mount Sinai Health System stands as one of New York's premier academic medical networks, employing 48,000 professionals across 7 hospitals, over 400 clinics, more than 600 labs, a nursing school, and leading medical and graduate education. They tackle complex health challenges by innovating science, treatments, and education, while nurturing local communities with top-notch care. Integrating hospitals, labs, and schools, Mount Sinai delivers holistic healthcare from infancy to old age, incorporating AI and data-driven methods centered on patients' physical and emotional well-being. The system includes about 9,000 physicians and 10 joint venture centers in New York City's 5 boroughs, Westchester, Long Island, and Florida. Their hospitals frequently top rankings from Newsweek's The World's Best Smart Hospitals, Best in State Hospitals, World Best Hospitals, and Best Specialty Hospitals, as well as U.S. News and World Report's Best Hospitals and Best Children's Hospitals. The Mount Sinai Hospital earned the U.S. News and World Report Best Hospitals Honor Roll for 2025 to 2026. For more details, check out https://www.mountsinai.org or follow Mount Sinai on Facebook, https://www.facebook.com/Mount Sinai NYCUTM medium equals CPC and UTM source equals Google and UTM content equals Google SEM and UTM campaign equals MSHS Respiratory Institute. Instagram, https://